Moving down into the stomach is really a storage tank. There isn't any absorption happening here, but there is release of gastric juices, which have lots of enzymes and acid that help to break down the food contents that have made it this far. So it's a storage site for the bolus of food that has been swallowed. It is about 50 mLs when it is empty and up to 1,000 mLs when it is full. So there's a lot of variability in the size of the stomach, depending on lots of different things like body habitus, um, your state, right? If you're elderly, if you're a child, that may be less. If you're an adult, maybe more. Things like your eating patterns can change the size and capacity of your stomach. And then these gastric juices that are released from these gastric pits. So the lumen and the mucosa of the stomach is thrown into these deep pits that we call gastric pits or gastric glands that have cells that line the glands that release, whether it be pepsinogen that can start to break down proteins or parietal cells that can secrete acid. These are going to release that gastric juice into the lumen of the stomach, and that will help with the movement and the mixing and the absorption of the contents in the stomach. Now, the anatomical parts of the stomach, we looked at this in the lab, so this will be kind of a review here, but the fundus being the top portion, the body being the middle portion, the antrum being the more distal parts, and the pylorus being the narrowed region that leads to the duodenum. The main role of the stomach is mixing chyme, right? The bolus of food that came through the esophagus is now much more liquefied as it reaches the stomach because of all the contents and the mucus and the enzymes being released. And so now it is a semi-solid mixture that we refer to as chyme. It is also going to be involved in gastric emptying. So that gastric emptying is going to be controlled by the pyloric sphincter. So that smooth muscle that guards the pylorus of the stomach and slowly releases this chyme a little bit at a time in a regulated fashion into the small intestine is what helps to regulate the pace of absorption. In terms of the inner lumen, the lining of the stomach is also expanded to accommodate many, not many, but large volumes of food and liquid. And so this creates what we call rugae. Rugae are the folds that increase the surface area of the stomach and allow it to be more expandable depending on different volumes of food content. Getting to the gastric pits are these depressions or these deep indentations of glands found in the stomach lining that release several different products. They're lined by cells, and the cells themselves are what release these products onto the lumen. So first off, pepsinogen. So pepsinogen is made by chief cells. And chief cells are these smaller yellow cells that are interspersed. They're not as large as the parietal cells, and they're going to be interspersed all over the gastric pits or gastric glands. Pepsinogen is going to be the precursor for pepsin. So when we release pepsinogen into the stomach lining, acid actually converts that pepsinogen into the active form pepsin, excuse me, and pepsin can begin to break down the proteins that are found in the stomach contents. We also have hydrogen ions or acid, hydrochloric acid being released by parietal cells. Parietal cells are much larger. They're not as abundant, and they're usually concentrated towards the base or the bottom of the pits or glands. The role of the parietal cells is to make the hydrogen ions, which creates the acidic environment that is important for breaking down food contents, also playing somewhat of an immune role, right? The acidity helps to defend against any pathogens, bacteria that may have ended up in the lumen of the stomach. Next, we have intrinsic factor. Intrinsic factor is required for the absorption of vitamin B12, and this is made by the parietal cells. And so parietal cells, again, are larger. They also make hydrogen ions. So there are two secretions coming from these larger, more basal located cells. And then we have G cells and mucus cells that are somewhat interspersed. And they're going to create, G cells make gastrin and mucus cells make mucus, right? These are also called neck cells. And the neck cells are what secrete mucus. They're usually found towards the neck or the proximal parts of the gastric glands. Now, the acidic environment in the stomach is about a pH of 2, so very acidic here, which is needed for activating pepsinogen, denaturing the proteins that are coming through in the lumen, and killing any bacteria or pathogens that have made their way from the oral cavity. This is where we have the gastric mucosal barrier. So the mucus that is secreted by the neck cells actually serves to combat some of that acidity. So that actually coats the lumen of the stomach and begins to neutralize some of that acid as it makes its way towards the antrum and even the pylorus and certainly into the small intestine. So the mucus creates this mucosal barrier that is made of mucus and bicarbonate that neutralizes the acid and coats the stomach wall against some of that acidity. Some of this is also made by goblet cells, by the way. Goblet cells are interspersed all throughout. So goblet cells are unicellular glands. There are glands that are made of one single cell. They're shaped like a goblet. We talked about them a bit in the respiratory system because they are in the respiratory tract as well. But they basically make mucus that is extremely important to the motility of the gastric contents as well. The small intestine. So small intestine is the next part of the tract leaving from the pylorus of the stomach. It begins at the duodenum, then into the jejunum, and then into the ileum. Again, parts that we have looked at in the lab. It is about 8 to 10 feet long, and it is the primary site of absorption mainly, but still a little bit of digestion still happening here as well. And if you think about the divisions of the intestine, we can think about the different parts being in different areas. So the duodenum is more so in the left quadrant, excuse me, upper right quadrant. Jejunum is in that upper left quadrant, and then ileum is in the lower right quadrant of the stomach, and then leading into the ascending colon. Some of the secretions that are released into the duodenum come from the pancreas and the liver. So the pancreatic juices enter through those duodenal papilla. We looked at some of those structures in the lab, the openings in the duodenum that allow those juices to be released. The juices are made of enzymes like lipase, amylase, etc., tryptase, and they are going to begin breaking down, not begin, but further breaking down and continue the absorption and digestion of contents coming through in the intestine. The other role of the pancreatic juices is to neutralize the acidity of the chyme. So there's a lot of bicarbonate in those juices to help to neutralize that chyme. The other juice or sort of content, secretions that are released into the duodenum is bile. Bile is made by the liver, stored in the gallbladder. We looked at the structures of the biliary tree in the lab. So the different hepatic ducts, the common bile duct that are going to release that bile, store it in the gallbladder, and the cystic duct, which is going to help to release it into the duodenum. And the role of bile is to break down fats specifically, a process called emulsification, where the fats are broken down to smaller components so that they can be more easily mobilized through the aqueous environment in the body. So the main role of the small intestine is absorption. And if you think about the entire length of that small intestine, right, about 8 to 10 feet, it is in the first 20% of that entire length that most of the absorption is completed. So there's not a whole lot of absorption happening throughout the rest of the length, but it is primarily occurring in the first 20%, really in the duodenum and a bit of the jejunum. In terms of its anatomical features and the way that it is arranged, first of all, it is tightly coiled up into the abdomen. If we were to open up the wall of the lumen, we would see that it is highly folded. The brush border, we talked about the brush border in the proximal convoluted tubule of the kidney. There is a brush border that exists here in the small intestine as well. And the role of the brush border similarly here is to increase the surface area to allow for absorption. So that happens through the villi and microvilli that are found in walls of the small intestines. All right. So kind of getting ahead of myself there, but the villi increase the surface area. It is the outpouchings or the folding of the lumen to create these kind of finger-like projections from the luminal wall. And then from those villi, we had even smaller projections called microvilli. So the cells of these um, villi are going to have their apical modifications called microvilli, which create that brush border, which is also an adaptation to help enhance absorption. If we think about the crypts of Lieberkruhn are the very similar to the gastric pits, but they're a different location here in the small intestine. But these are the deep pits or crypts that the lumen of the small intestine is folded into to help increase the secretion of bicarbonate-rich fluid that is also working to neutralize the acidity of the chyme coming from the stomach. So there's a release of uh, um, not gastric, but the pancreatic juices into the stomach. There's a release of other secretions into the small intestine. Did I say stomach? Excuse me. Pancreatic secretions are released into the small intestine, not the stomach. And then the absorption is also being facilitated by the villi. Okay. So here are the villi. Here are the crypts of Lieberkruhn, again, where these bicarbonate-rich secretions are made and released. And then the villi themselves. And then on the apical surface of the cells of the villi, we have the microvilli making the brush border. Now, in the middle of the villi, we also have the arteries, the veins, and then these lymphatic structures called lacteals. We'll talk about what these are a little bit coming up, but the lacteals are really important for the absorption of fats. They are connected to the lymphatic vessels and lymphatic ducts, and these allow fats to find an alternate route to get into the plasma. Fats are sometimes too large and bulky to cross right into the plasma. So they are first absorbed into the lymphatic system, then they circulate in the lymphatic ducts and vessels. Then they are dumped into the venous system, and then they can enter the circulatory system, kind of like an alternate route to get those fats to be absorbed. Now, in terms of the liver also plays a role in absorption because of the portal system. The liver receives all the nutrients that is absorbed into the bloodstream through the intestinal organs, starting at the stomach, including the small intestines and any parts of the large intestines. And it brings all of those nutrients through the portal system, through the mesenteric veins and the splenic vein, into the hepatic portal vein, and then finally into the liver sinusoids. Here, the liver plays a role in detoxifying any substances that are being absorbed and processing, sorting any nutrients that are also being absorbed. And then that blood can be returned into the venous system via the hepatic veins, which make their way to the vena cava. So the liver plays not only a role in terms of the bile that is being made, but it plays a direct role in the absorption of the nutrients from all parts of the GI tract. Okay, so kind of reiterating that here as well, the nutrients that are being released in, or not released, absorbed in the small intestine, the stomach, etc., are making their way into the hepatic portal vein. And then these nutrients, including electrolytes and water, will go into the hepatic circulation and then eventually into the back into the systemic circulation. Moving into the large intestine, so this is made up of the colon, the cecum, the rectum, and then it's leading into the anus, right? The external opening. So this is mainly involving or starting at the ileocecal junction between the ileum, the smallest distal part of the small intestine, the ascending colon, which really starts at the cecum. Here we also have the appendix, which we talked about, ascending colon, transverse colon, descending colon, sigmoid colon, and then ending at the rectum. The colon and the large intestine, their main role is for contraction of that waste into feces and elimination of that final waste product, and then reabsorption of any water that is still present in the lumen at this point. It's also going to store that feces until defecation. Okay. If we think about the rectum and anus, these are the very distal most parts of the tract. They start basically where the colon ends, or the sigmoid colon more specifically ends. And it's basically the sphincters at this point. The internal anal sphincter and then the external anal sphincter are sort of regulating the flow of feces based upon the nervous system and based upon the amount of feces present in the large intestine. And then these sphincters are differing again, kind of like the upper esophageal sphincters. The smooth muscle is making up the internal sphincter, so involuntarily controlled. And then the external sphincter is made up of skeletal muscle. So we can control that and decide when we'd like to void or go to the restroom under our voluntary control. And relaxation of both sphincters is what's required for defecation. So there needs to be sort of a collaboration or synchronized release of the internal sphincter and then the voluntary release of the external. Shifting into some of the accessory glands here. So the salivary glands, these are found in the oral cavity. There are three different groups of salivary glands: the parotid glands, which are the largest, the submandibular glands, and then the sublingual glands, which we've talked about when we look at the oral cavity in the lab. And their main role is to make saliva. The pancreatic gland, so remember the pancreas has an endocrine part, the islets of Langerhans, and then it has the exocrine part, which is made of its acini. The acini are the glandular bulbs or the glandular cells that are making these secretions that are released into ducts, hence the exocrine part of the gland. And then finally, the liver. The liver also has its role in terms of making bile. The liver is actually the largest gland in the body, and it releases its secretion, bile, into its separate ducts, which form the biliary tree. So this is how acinar structures, and remember, all glands are made of epithelial cells. So epithelial cells have two roles. They form our body cavities, right? Like the tract, making the epithelial lumen, the skin, the lining of all of our tracts are epithelia. And the second major role of epithelial cells is to make glands. So these acini are epithelial cells that come together. They have a specific function in terms of what they will secrete, and they are forming acini. So we have several different types of acini: liver acini, pancreatic acini, salivary gland acini. The acini make the secretions and release them into the ducts, and then they are basically fine-tuned or modified by other ductal cells. The cells that are lining the duct itself, also epithelial cells, can release certain enzymes, can release certain ions, can release water or sometimes mucus that can alter the consistency or the overall makeup of that secretion before it is finally released onto the lumen of whatever structure it's being released, whether it's oral cavity, small intestine, etc. Some of the characteristics of saliva. So saliva is bicarbonate rich. It contains mucus for lubrication. And then it also has salivary amylase to help begin the breakdown of starch and lysozymes to play a defense role, right, to immune or launch an immune response against any pathogens or bacteria. Moving on to the pancreas also has exocrine and endocrine portions. This is things we've discussed before. The exocrine portion release the pancreatic juices, which is a cocktail of juices involving bicarbonate to help neutralize the chyme, pancreatic enzymes like amylase, lipase, proteases, and nucleases that can begin to break down these relative biomolecules. So amylase and lipase break down starch and fats, proteases break down proteins, nucleases, excuse me, break down nucleic acids. In terms of the weight of the pancreas, in terms of its size, it secretes much more protein than any other tissue in the body. All of the various enzymes that it secretes, these are all proteins. So it is one of the most overworked gland in terms of the amount of protein that it secretes, breaking down many different types of biomolecules, and then creating that cocktail of pancreatic juice that also has bicarbonate in there as well. The liver, very similarly, can secrete bile, and then it also has a role in processing nutrients, so it can help us undergo glycolysis, the breakdown of not glycolysis, excuse me, glycogenolysis, which is I'm making I'm blanking on the term here. Forgive me, guys, but glucose to glycogen. So basically the storage of that glucose to glycogen, the opposite of glycolysis. It also helps us to store away amino acids and fatty acids and convert those into things like proteins or even larger peptides, as well as triglycerides and cholesterol. And so the liver has a lot of different roles in terms of our fed and fasted state, right? Things like the insulin response versus the glucagon response and how we can turn on and off those two different regulatory pathways. So a lot of varying functions with the liver. Other functions include removal of old erythrocytes, so it can sort of sequester those dying erythrocytes, break them down into their heme and globin, and sometimes recycle those components. It generates bilirubin, and then it eliminates toxins, right? So bilirubin helps us to eliminate various toxins and bile pigments, which is kind of the end form of that bile, the breakdown components of that bile. And then other structures like plasma proteins are made in the liver. This helps with clotting, like the clotting of blood, albumin, oncotic forces that we've talked about that help to maintain the hydrostatic forces in our blood pressure, and then modification of hormones. A lot of different hormones that we've looked at throughout the entirety of this course, like the renin-angiotensin system. Renin is made in the liver as well, clotting factors. All of these are made and released from the liver.